patient of having a pus we have received in the lab, and the result was few gram positive cocci arranged singly and in pairs. Okay, so this gram positive and gram negative thing you will learn later on in the other practical sessions. But here you can see only that gram positive cocci and the arrangement of that particular organism. Like some of the cocci are arranged singly, some of the cocci are arranged in pairs. The third report of sputum sample, and in that sample we have found plenty of gram positive pairs and short chains. So in this particular sample, we have found two kinds of, and see the arrangement of the ground, so see specimen. Okay. Now you are all aware about the kingdom Protista. Okay. So prokaryotes and eukaryotes, these are not the new words for you. And in the prokaryotes, there are two actually. Just give me a 1 second. Sorry. So in prokaryotes, the two categories, bacteria and blue-green algae, and in eukaryotes, the algae, protozoa, and fungi. Okay, this is the basic difference of prokaryotes and eukaryotes. Starting with the size, prokaryotes ranges from 1 to 10 micrometer and eukaryotes ranges from 10 to 100 micrometer. The complexity of prokaryotes, like they are unicellular structures, and eukaryotes are multicellular structures. Now in prokaryotes, we never cytoplasm. But in case of eukaryotes, you will find mitochondria, lysosomes, endoplasmic reticulum, and other organelles. In prokaryotes, the nucleus is diffuse, and in eukaryotes, the nucleus is present and it is well defined. The nuclear membrane, nucleolus, and ribonucleoprotein is absent in prokaryotes, and it is present in eukaryotes. The chromosomes, they are found in single and circular morphology in prokaryotes, and in eukaryotes, they are found usually several and in linear. In prokaryotes, the nuclear division was by binary fission, is by binary fission, and in eukaryotes, it is by mitosis or meiosis. The muramic acid is present in prokaryotes and it is absent in eukaryotes. Sterols are absent in prokaryotes and present in eukaryotes. Extrachromosomal DNA, it is found in plasmid, and in eukaryotes, it is found in mitochondria. Now, the size of the bacteria, it is expressed in terms of micron or micrometer. And the medically important bacteria, they are ranges from 225 into 0.2 to 1.2 micrometer. These are the various shapes of bacteria. One with circular, or we can say the oval, somewhat oval structure, it is known as coccus, and the rod shape structures are known as bacillus. These are the two important shapes of bacteria. So the major groups of bacteria on morphological basis, they are divided into 5 categories. First is true bacteria, second actinomycetes, third spirochetes, fourth mycoplasma, 5 rickettsia and chlamydia. So starting with the true bacteria, they are rigid, unbranched and free living forms, and basically they are divided into 2 shapes. Cocci, they are spherical or oval cells, and bacilli, they are rod shaped cells. Again, this is the slide with the arrangement and shapes of bacteria. Here you can see the 2 cocci, they are placed side by side, and they are known as diplococci. And this kind of arrangement is seen with the Streptococcus pneumoniae. Just like that, the cocci, they are arranged in clusters in case of Staphylococcus aureus. So there are other arrangements are also seen like in tetrads, in Sarcina, and some of the cocci, they are arranged in chains. Basically, it is seen with the Streptococcus pyogenes. Bacilli, if they are arranged in chains, it is seen in Bacillus anthracis. Now, there is a flagella which is arranged in different poles or surrounding whole of the bacteria in different kinds of bacteria. Like in Vibrio cholerae, the one single polar flagellum is present. And in case of Helicobacter pylori, the tuft of flagella is present at one pole. So starting with the cocci, they are spherical or oval bacteria arrangements. So this is diplococci, the cocci arranged in chains or long chains, or they may be arranged in grape-like clusters. These are the examples of organism. The cocci, which are arranged in grape-like clusters, it is seen with the Staphylococcus group. In Streptococci, they are arranged in long chains, and in Pneumococci, they are seen as diplococci. Okay, so this is a diplococcus and this is a chain cluster. Okay, now switch to bacilli. They are rod-shaped bacteria and they divide in one plane. Cocobacilli, the length of the organism is approximate its width, and it is seen with the Brucella species. Streptobacilli, it is arranged in chains, and example is the Streptobacillus monoliformis. Now, some of the bacilli, they are arranged at angles to each other, and it is known as Chinese letter pattern of the arrangement. And it is particularly characteristic of Cornybacterium diphtheria. Now, Vibrio cholerae, the bacilli, they are curved shaped or coma shaped. Then actinomycetes. They are rigid organisms like true bacteria, but they resemble fungi because they exhibit branching and filaments, like you can see in this picture. The spirochetes, they are longer and having several coils in its morphology. Mycoplasma, they lack rigid cell wall. So they can change their size and shape accordingly, and that's why they are pleomorphic. Pleomorphism means the organism can change their shape or size. The round or oval bodies, and they interlacing filaments is present. Rickettsia and chlamydia, they are very small and they are obligately intracellular and they are closely related to the virus. Now, starting with the bacterial anatomy that is universal to all the bacteria, that they possess cell wall, plasma membrane, cytoplasm, nucleus, flagella, pili or fimbria, capsule, intracytoplasmic inclusions, and spores. Here you can see the outermost layer is the capsule. Then there is a cell wall. Then there is a cytoplasmic membrane. And underneath the cytoplasmic membrane, there is a cytoplasm and the nuclear material. Okay. This is the pili and this one is the flagella. So starting with the cell wall is actually tough and rigid structure that surrounds the bacterium like a shell, and its thickness varies between 10 to 25 nanometer, and it possesses 20 to 25% of dry. Does not stain with the simple stains like gram stain. Whenever you do gram stain, if the sample possesses the bacteria having capsules, then you can see the clear halo because the capsule does not take the stain. Cannot be seen by direct light microscopy, and the chemical structure of the cell wall of gram positive bacteria, it is simpler than the gram negative bacteria. Now, functions of the cell wall: it provides the characteristic shape to the cell, it confers structural rigidity, it protects the bacteria from changes in the osmotic pressure, protects the delicate underlying cytoplasmic membrane, and maintain the high intracellular level of metabolites. It takes part in cell division, provides a rigid platform for surface appendages like flagella, fimbria, and villi. It provides attachment sites for antibiotics and bacteriophages. It is made up of peptidoglycan polymer, okay, maybe mucopeptide or murine, sugars like N-acetylglucosamine and N-acetylmuramic acid table, and the D and L form of amino acids. So this is the basic structure of the cell wall. Here you can see the alternating sequence of N-acetylglucosamine and N-acetylmuramic acid, and this each peptidoglycan layer connected or cross-linked to other by a bridge made up of amino acids or amino acid derivatives. Now, the cell walls of all bacteria are not identical. The cell wall composition, it is an important factor in bacterial species analysis and differentiation. There are two major types of wall, the gram positive and gram negative. So the cell wall of gram positive bacteria, it is simple chemical nature, and it contains two characteristic uh, molecules known as the peptidoglycan or teichoic acid, which is different than gram negative bacteria. So the peptidoglycan, it is a thick multi-layered exterior to the cytoplasmic membrane, and many polymer layers of peptidoglycan, they are connected by amino acid bridges. So teichoic acid, and other thing is the teichoic acid, it is a polymer of polyribotyl phosphate and polyglycerol phosphate, and it is a major surface antigen. Okay. Here you can see the structure. This is the cytoplasmic membrane, and this one, this blue color is the peptidoglycan layer, and this one is the teichoic acid. Teichoic acid is the characteristic of gram positive bacteria, and it is not found in gram negative bacteria. This peptidoglycan layer is also different in case of gram negative bacteria. So the cell wall of gram negative bacteria is complex. It is the peptidoglycan layer is much thinner compared to the gram positive bacteria. There are two membranes present, the inner cytoplasmic membrane and the outer membrane. So here you can see the peptidoglycan layer, it is very thin compared to gram positive bacteria, and there is no presence of teichoic acid that was present in gram positive bacteria. Here you can see the LPS layer that is characteristic for gram negative bacteria and the pore proteins. Now the periplasmic space, okay, here you can see the periplasmic space. It is distinct space between the cell membrane and the outer membrane, and it contains degradative enzymes. It destroys potentially dangerous foreign matter present in this space, and specific binding and transport protein for amino acid. Outer membrane. Okay. Now this is the structure of cell wall of gram negative bacteria. This is the LPS layer or the lipopolysaccharide layer of gram negative bacteria. It is a high molecular weight complex glycolipids and contains three regions. In region 1, there is a polysaccharide portion and that determines the O antigen specificity. The region 2 contains the core polysaccharides, and region 3, it contains lipid A. Now this lipid A, it is the integral part of outer membrane, and it is also known as endotoxin, which is characteristic of gram negative bacteria. It is not present in gram positive bacteria. And it is effects associated with harmful gram negative bacteria. Outer membrane proteins, as you have seen in this slide, the porine channels. Okay, so they form the transmembrane pores, and they permit the passive diffusion of low molecular weight hydrophilic compounds, sugars, amino acid, and certain ions. And it serves as a specific receptor for some bacteriophages. So this is the real picture of gram negative bacteria. Here you can see the gram negative rods. Okay, they are the gram negative bacilli, and surrounding it you can see the clear halo, and that is the capsule of this particular gram negative organism, which does not take the stain. Okay, this is the picture of gram positive bacilli. This one is the gram positive cocci. Okay, they are arranged in pairs. Some of the cocci are arranged in chains, and this one is the gram negative cocci. Right now, you just remember the gram negative cocci will appear pink in color, and the gram positive thing will appear blue in color. The reason you will understand in the practical of gram stain. Now, cytoplasmic membrane. It is a thin elastic semi-permeable layer that lies beneath the cell wall, and it's separated from the cell cytoplasm. Chemically, this cytoplasmic membrane is consists of phospholipid bilayer with small amount of carbohydrates. Sterols absent except in mycoplasma. Function, just I have mentioned in the earlier slide that it is a semi-permeable membrane, and inward and outward passage of water and small molecular lipid soluble solutes takes place by diffusion through this membrane. It contains enzymes that is involved in cellular respiration, synthesis of cell wall and outer cytochrome oxidase coating purpose present in cytoplasmic membrane. Now, cytoplasm, it is a colloidal system containing organic and inorganic solutes in the viscous watery solution of the bacteria. First organelle which is we will discuss is ribosome. It is a small electron particle and integrated in linear strands of mRNA. It is slightly smaller than eukaryotic cells, and sedimentation rate is 70 S. The protein synthesis site, the function of the ribosome is protein synthesis, and it is a site of action of several antibiotics like aminoglycoside, macrolide, and tetracyclines that you will learn. Mesosomes, they are the colloid invaginations of cytoplasmic membrane that extend into the cytoplasm. They are vesicular, convoluted, and multi-laminated structures. They are more prominent in gram-positive bacteria, and they are the main sites of respiratory enzymes, or they are the sites of synthesis of cross-wall septa. Intracytoplasmic inclusions, accumulation of food reserves like polysaccharide, lipid, or phosphate, polyphosphates occurred in this intracytoplasmic inclusions. They are sources of carbon or other inorganic substances, and they are the characteristic of different species. Volutin granules, or the metachromatic or Webb-Ernst granules, which is characteristically seen only and only in Corynebacterium species. They are highly refractive, strongly basophilic bodies consisting of polymetaphosphate, and they represent a reserve of energy and phosphate for cell metabolism. Here you can see these are the granules at the end of the bacilli. They are known as volutin granules or metachromatic granules or Babe-Ernst granules. And it is particularly seen in Corynebacterium species only. Albert staining is useful to see this volutin granules, and they will appear bluish black or greenish in color. Now, nucleus. So there is no true nucleus present, and there is no nuclear membrane or no nucleolus. The genome which is present in the bacteria is single, circular, or double-stranded DNA. There is some extra nuclear genetic element is also present in some of the bacteria, and they are known as plasmids. So they are transferred from one bacterium to another bacterium either through conjugation or through bacteriophages. This conjugation and bacteriophage that you will learn in subsequent lectures. They confer properties like toxigenicity to that particular organism or like drug resistance to that particular organism. They are mostly found in gram-negative bacteria. Now we will talk about capsule and the slime layer. So capsule is the amorphous viscid bacterial secretion deposited outside the cell wall, and it is very sharply defined. Here are some of the examples of some bacteria like Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenzae, and Bacillus anthracis, and they possess this capsule. Now, what is the difference between this capsule and the slime layer? Here you can see that slime layer, or we can say the biofilm, it is less discrete structure or matrix which embeds in the cell. And it is not as rigid and clearly demarcated compared to capsule. So here are some of the methods by which we can demonstrate the capsules of the bacteria, like in gram stain, because this capsule will not take up this stain. So you will see the unstained halo around the bacterial body. Here you can clearly see there is unstained halo, that is a capsule. In this picture, you might not see clearly, but in this picture, you can see clearly the capsule. Another method is the negative staining or the India ink or Nigrosin staining, in which you can see the clear halo around the bacterium against the dark background. Okay, so this is the background, this is the cell, and this one unstained portion is the capsule of that particular organism. The another method is the Quellung reaction or the immunological method to detect capsule of the organism, in which the suspension of the capsulated bacteria will be taken, and some specific antiserum is added to that suspension. So what happens, the antigen antibody reaction occurs and the capsule become delineated, refractile and swollen, and you can see such kind of reaction under the microscope due to the increase in its refractivity. Okay, so this one is the Quellung reaction, or we can say the capsule swelling reaction. It is useful for typing of the bacteria. Function of the capsule, it protects the bacterial cell from the engulfment by the protozoa or the phagocytes or white blood cells. It protects from attack by antimicrobial agents and enhance the bacterial virulence because it cannot be destroyed by the body's immune cells. It protects cell from effect of drying or desiccation, protect the cell from degrading enzymes and bacteriophages, and help in cell adhesion to host epithelial cell surfaces. So we'll just end here, and the next topics we'll cover in the next lecture of morphology. So this is all. Thank you.